Okay, I guess it's noon here in New Haven, so it's my pleasure to welcome you all to a new global IMMUNOTALK. I'll be very brief, just to remind you that we are here and hopefully trying to achieve the goals that we have set forth, which are to benefit and inspire immunologists across the world in an egalitarian manner through these incredible talks, and to increase the opportunities for scientific learning without traveling. As we always say, we could not do this without the fantastic contribution of our speakers. We have Zaza Ndhlovu today, who is going to be introduced very soon by Elena. We're very excited to have him today with us. And we will have Richard Loxley next week. So stay tuned, and we will soon have some more news for the coming year. So hopefully you stay tuned for that too. So with this, I would love to introduce Elena, who will introduce our speaker today.
Perfect. Thank you so much, Carly. So it is a great pleasure and very, very special to introduce to you today Dr. Zaza Ndhlovu. Zaza is native from Zambia, and he is currently based in Durban, South Africa. He received his bachelor degree from Nelson Mandela University, and in 2005, he moved to the US to perform a master and doctoral thesis at Johns Hopkins University. And afterwards, a postdoctoral training at the Massachusetts General Hospital at Harvard University. In 2012, Zaza was appointed to a junior faculty position at Harvard. And it was then that he decided to set up his laboratory and research program at the University of KwaZulu-Natal in Nelson Mandela School of Medicine at Durban, South Africa. And I think we will learn more about that decision later during the question. Zaza has since then been associated with both institutions in US and South Africa, and he's currently an associate professor at Africa Health Research Institute and University of KwaZulu-Natal, as well as an assistant professor at Reagan Institute of Massachusetts General Hospital at Harvard. So Zaza's research has always been focused on T-cell responses in the context of viral infection, and I think nobody will question about the relevance of this important topic. During his graduate studies at Johns Hopkins University, Zaza used measles virus infection and vaccination as a model for defining features of effective cell-mediated immune responses to a virus that is successfully cleared. And he discovered then that virus-specific cells were heterogeneous in their functionality, and that functional features were depending on the molecular features of the stimulating antigen-presenting cells. When he transitioned for a postdoctoral training under the supervision of Dr. Bruce Walker at Harvard University, Zaza shifted his focus to HIV infection, and since then he has continued studying HIV infection. So then, in an effort to understand how a vaccine-induced T-cell response could control viral replication in the absence of sterilizing immunity, he studied long-term non-progressors and HIV elite controlling, yielding extremely important insights into the mechanistic differences in CD8 T-cell functionality between people who control HIV infections and those who have progressive infection. So Zaza's current lab in Durban, South Africa continues focusing on HIV as well as on mucosal and lymphoid tissue immunology. Among different areas of HIV immune responses, Zaza and his team investigates the acute phase of HIV infection to understand initial immunological events that contribute to immune dysfunction and subsequent disease progression. And we will learn more about this topic during his talk today, and I cannot wait for that. Zaza's significant research work has been published in top-tier journals, And he has received many accolades, including in 2017, the very competitive Howard Hughes Medical Institute International Research Scholar Award. So Zaza, it's a great honor and a great pleasure to host you today as a global immuno speaker. We are deeply thankful for all the efforts you made to participate and presenting today. We really appreciate it.
Thank you.
As you know, the format of this Global IMMUNOTALK, we like to ask a question to our speakers. And I think that your career is very interesting. You had a faculty position at Harvard, one of the top universities in the world. And you decided to go back to South Africa to establish your own independent program. So the question we have for you today is, what motivated you to go back to Africa and establish your independent laboratory in South Africa? And what are the advantages and disadvantages related to that decision?
Thank you very much, Elena, for that introduction. Thank you for having me. And I'm very honored to be able to share some of my work here. I think for me, it was a very easy decision to make. First of all, I'm an African, and I really always wanted to come and contribute to African science. Secondly, I was an HIV immunologist and studying HIV, and Africa has the largest epidemic, and South Africa happens to have the largest epidemic. And then the reason I chose to come to South Africa, not my native country, Zambia, is that South Africa has the infrastructure to do basic science, almost the level that I was doing at Harvard. So, and the fact that I also retained my affiliation with Harvard, I think it was an easy choice for decision for me to make, because not only can I do science here on site and work with humans, but I can also contribute to training the next generation of African scientists, and that's what I've done. And so I've recruited a lot of students, I've graduated PhD students, postdocs, and I have a much bigger footprint here in South Africa than I would have had if I'd stayed in the US.
That's fantastic, and we really appreciate that you are training the next generation of scientists. In South Africa.
Yeah, by the way, 90% of my lab is female, so they're mostly women in my lab. So that's another.
Thank you so much, Zaza. Thank you so much. You have our admiration, really. So we are looking forward to your talk, and if you wish, you can share your screen.
Okay, let me go ahead and share my screen now. All right, so today, I'm honored to give this presentation about my work, and I had two slides kind of giving a little bit of background about myself, but Elena has done justice to that, so I'm not gonna go too much into that. But suffice to say that I'm Zambian. When I graduated from college, I started working at the University Teaching Hospital in Lusaka, Zambia, and then I moved to Hopkins for my graduate studies, and to Harvard and Mass General for my postdoctoral fellowship, and back to Africa for where I set up my lab. And like Elena said, I have affiliations with the Reagan Institute, ARI, which is Africa Health Research Institute, and the University of KwaZulu-Natal. So my talk today will be in 4 parts. First of all, I'll tell you about using hyperacute HIV infection cohort for HIV cure studies. I'll define what we mean by hyperacute HIV as we go along. Then I'll tell you a little bit about very early initiation of antiretroviral therapy, how that improves immune function and accelerates HIV reservoir decay kinetics. And then I'll go on to tell you a little bit about some of the work, and I'll show you some of the work that we are doing trying to define the HIV reservoir and the HIV immune microenvironment in lymphoid tissues as a way of contributing to developing a cure for HIV. And lastly, I'll talk very, very briefly about some of our CD8 T-cell work, trying to understand why CD8 T-cells fail to accumulate in lymphoid tissue follicles, and some of the efforts we're doing to reverse that. So I just want to start by emphasizing that we can easily forget about HIV given the COVID-19 pandemic that has just ravaged the world. But HIV still remains a very significant global pandemic, with 37 million people living with HIV globally. And 50% of those people live in sub-Saharan Africa. And that's clearly the motivation for why I decided to come back here, because that's where the problem is. And we have more than 17 million people on lifelong antiretroviral therapy, and South Africa happens to have the largest antiretroviral therapy program in the world. But in spite of all these intervention efforts, we still have more than 2 million new cases occurring every year. So the problem is still very much with us. So let me tell you a little bit about KwaZulu-Natal and Durban, where we work. So KwaZulu-Natal is a province of South Africa on the east coast of South Africa on the Indian Ocean. And Durban happens to be a city just by the Indian Ocean, which is a very, very beautiful city. Throughout the year, it's beach season. So it's a wonderful, wonderful place to visit if you ever get a chance. But some of the surrounding areas around Durban are highly impoverished, just like most of the African countries. And Umlazi is a big township where we work from. It's the second largest township in South Africa, only second to Soweto, where it's densely populated and has, unfortunately, very high HIV prevalence. So just here, just the data just showing you HIV prevalence in South Africa, and KwaZulu-Natal has the highest HIV prevalence in the country. So South Africa itself is the epicenter, and KZN is the epicenter of South Africa. So we're really at the epicenter of the HIV epidemic. So, our efforts have been trying to see how we can contribute towards a cure for HIV. We know that a cure is a very ambitious undertaking. It's extremely difficult to cure HIV, but there's proof that actually it can be done. And an example is the famous Berlin patient Timothy Brown that received a bone marrow transplant, and the virus was eradicated for a very long time. Unfortunately, he passed away from his cancer complications earlier this year. And then recently, the London patient also was underwent a similar procedure and was cured. At least for now, there is no virus detectable. Although these are not public health measures that can be rolled out to scale, I think they're proof of a principle that actually a cure is actually possible. But it's also been shown that very early initiation of antiretroviral therapy can actually, in some special instances, link to at least prolonged remission of therapy. And this was demonstrated many years ago now by the Visconti cohort, where individuals were put on therapy and remained in remission for more than 10 years. And in the US, we heard of the Mississippi baby. This is a baby that initiated antiretroviral therapy at birth, and the baby went on into remission for at least 28 months. And since then, there's been a lot of examples. And here in South Africa, we have, like I said, we have very high HIV incidence. These are data that we generated from Northern KwaZulu-Natal, most recently by our group here at Africa Health Research Institute, where you can see that HIV prevalence among young women between 27 and 44 is greater than 60% compared to their male counterparts. And so it's easier to actually identify acute HIV infection in such a setting. And that's why we set up a cohort that we refer to as FRESH. And FRESH stands for Females Rising Through Education, Support, and Health. And this cohort has two major goals. One is HIV prevention and poverty reduction, and the other goal is immunology, studying the immunology and virology of this hyperacute HIV infection. So what we do in this cohort is that we identify young women who are profiled to be at high risk of HIV acquisition, but at the time that they are recruited, they are HIV uninfected. they come to our center for classes. They take a course where they learn about HIV prevention. They learn about, you know, skills, to get jobs, and computer skills and other skills that would help them to empower them economically. At the same time, we test them for HIV by a finger prick blood draw to detect HIV RNA, and we collect samples every once a while. And once they become HIV infected, then we lump up sample collection. And so far, using this strategy, we have identified 90 acutes, and most of them we have identified them in the earlier stage of HIV infection, which is referred to as HIV phobic stage 1. Besides that, like I said, we also try to provide as much HIV prevention information as we can, and PrEP is one of the programs that was introduced recently, and our uptake was very, very poor when PrEP was introduced in Fresh, but now has improved quite significantly. But maintenance of girls on PrEP has always been a challenge. People take the first ones and then they stop taking PrEP. But more recently, we are very, very encouraged by the interim analysis from the HPTN of 084 study, where it was shown that a long-acting injectable cabotegravir administered every 2 months was 89% more effective than a daily pill in preventing HIV acquisition in women. We believe this will be a game changer in terms of the epidemic, particularly in this very hard-hit demographic of young women. So from acute HIV infection, what can we learn from that? Why do we want to study acute HIV infection? So there are 4 key questions that we'll focus on. One is we want to characterize the transmitter founder virus. We know that HIV is initiated by a single transmitter founder virus, and if we understand that, we can interfere with HIV transmission. We want to understand the initial immune responses to HIV infection and why these responses ultimately fail to clear the virus. We also are interested in trying to understand the reservoir seeding dynamics and how we can interfere with the reservoir. And we also want to really understand, you know, good immunity from ineffective immune responses that can inform vaccine design. So in Fresh, we've done several studies and that are unique and answer important questions that otherwise would not be answered by other studies. The fact that we have samples prior to somebody getting infected provides us with opportunities, first of all, to understand risk of HIV acquisition. Because you have a group of girls that have the same risk profile, and some of them become HIV infected and most of them do not become HIV infected, you can ask the question, what is unique about the individuals that become HIV infected? And this is a study that was done in Fresh, led by Daquan, where they looked at the girls that became infected in Fresh versus those that did not become infected, and found that the microbiome in the vagina played a major role. So if you have a microbiome that is dominated by lactobacillus, you are less likely to acquire HIV than if you have a mixed flora of bacteria in the vagina microflora. And this was shown that to be significant, and these data were published, and there are studies now underway to try and intervene and shift the microbiome as the strategy to prevent HIV acquisition. Another study that we did earlier on when we just started Fresh, that was led by me when I just moved to South Africa, was really to try and understand the initial immune response to HIV. In this arrow where I'm showing you, this is the phase during the upswing in viremia that we refer to as hyperacute HIV infection. So what happens to the initial immune response to HIV? What we found, and these are published data, so I'm not going to go into too much detail, is that we were surprised at how big the immune response is, at least the CD8 T cell part of it. There was a massive CD8 T cell response to an extent that almost 60 to 70% of all circulating CD8 T cells were activated and specific for HIV in the individual. The reason why there is a response, this response doesn't clear the virus, is that this response had defects. Because it was an exaggerated response, it had suboptimal antiviral activity in terms of interferon gamma secretion when you compare to CMV at the same time. And also these cells were highly apoptotic and that didn't last for a long time. They all died before they finished the job. And these data were published almost 6 years ago, if you want to know the details. Then in the cohort, the fresh cohort, the early treatment was introduced, immediate therapy, because it was shown by us and others that in treating HIV early has clear benefits than delaying treatment. And so when individuals initially, when we started, we were treating according to the South African treatment guidelines. You can see the viral load picking up very rapidly and going to a very high peak and then coming to a set point, and then the CD4 going into a nadir and rebounding slightly. When you treat people early in phoebic stage 1, this is where you detect HIV RNA, and within 24 hours, you ask them to come back and they're put on therapy. You can see the peak viremia is blunted by several logs, and you preserve the CD4 T cell count. And if you can see this, these are summary slides. These data were published in Lancet a few years back now. You can see that you can blunt peak viremia by several logs. What was surprising is that when you measure total DNA, which is a crude way of measuring the reservoir, we did not see a difference. This was not really surprising, given that we were already known that the reservoir is established very, very early. But what is really reassuring is that when you follow people up and measure total DNA in these individuals that initiated therapy in phoebic stage 1 over 1 year period, you can see there's a significant reduction in total DNA. We've done some other sophisticated assays now that demonstrate that the reservoir decay is much, much rapid if you initiated therapy very early, compared to untreated infection where the reservoir tends to be very stable over a long period of time. So we were interested in understanding what are you doing to the immune system when you treat people this early. And so we wanted to compare immune responses in individuals treated in phoebic stage 1 versus people treated in phoebic stage 3 and 5 or untreated individuals. And here just looking at HIV-specific CD8 T cells measured by tetramers. And you can see that we were able to detect HIV-specific CD8 T cells with tetramers, although it was significantly reduced in terms of magnitude compared to untreated infection. But then we wanted to see what is really the driver of the HIV-specific response. And what we found is that it's the amount of antigen that they're exposed to before you suppress HIV that determines the magnitude of the response. In this, we measured what we call viremia copy days. This is taking into account peak viremia and the amount of virus, the days it takes for the virus to be completely suppressed in plasma. If you take that as a measure and compare that to the magnitude, you see that there's a correlation there. So then we went on to look at the quality of the immune response, starting with the CD8 T cells, because we had studied the CD8 T cells in untreated infection. And we had seen that when you stimulate them ex vivo and look for interferon gamma here by flow, you find that a very small fraction of tetramer-positive cells secrete interferon gamma, suggesting that they were defective in this particular function. When we compare to people treated in phoebic stage 1 to 3, in the same assay, stimulated and stain with a tetramer and look for interferon gamma secreting tetramer-positive cells, here you can see that there's a significant increase in tetramer-positive cells secreting interferon gamma, suggesting that this defect is actually reversed when you treat people early. We also looked at CD4 T cells. So not only do you preserve CD4 T cells when you treat people early, but the quality of the CD4 T cell function is preserved. Here we looked at CD4 T cell proliferation by CFSC dilution at 14 days and after 1 year, and you can see that the CD4 T cells were able to proliferate when you stimulate them ex vivo from individuals that were treated in phoebic stage 1. This is in a sharp contrast to when you treat people in untreated infection, where not only do you lose a lot of CD4 T cells from just direct infection, but the quality of the remaining CD4 T cells is compromised. Here you can see the CFSC dilution in the same assay, very few cells CD4 cells were able to proliferate, and even over time, you lose the ability of CD4 T cells to proliferate in an in vitro assay. So we went one step further and wanted to look at the phenotypic characteristics of HIV-specific CD8 T cells in treated, early treated, or untreated individuals. Here, again, I'm looking at tetramer-positive cells and looking to see, their differentiation profile. You can see that in early treated infection, the CD8 T cells are able to differentiate fully and become effector memory cells. Whereas when you delay treatment, you can see that the differentiation is arrested in transitional memory. And this is very similar to what has been published by others as well, that in HIV infection, the cells, the memory cells, HIV-specific cells are mostly transitional memory cells. And then we followed these individuals up for about a year and again looked at the same responses. And again, what we saw that in phoebic stage 1, these are tetramer-positive cells overlaid on total CD8s. You can see that mostly they differentiate and become central memory CD8 T cells. Whereas if you delay treatment, you can see that most of the cells do not become central memory. And when you look at untreated infection, the cells are really arrested in the transitional memory phase and they don't differentiate fully. So the early events that happen, there's an imprint in these CD8 T cells and they don't change over a long period of time. That's clearly showing that, potentially when you treat people early, they have the potential to generate memory responses, which if you stimulate it with a vaccine, you challenge it with a vaccine, they would have better responses. So to conclude on this part, I've shown you that natural HIV infection induces this massive defective immune response, and early treatment does augment immune function and induces memory responses. And I've also shown you that immediate therapy accelerates HIV reservoir decay kinetics. So the next part of my talk, I'm going to talk about some of the work that we're doing now in the lymph node tissues, trying to understand HIV reservoir and immune responses in lymph node tissues. So it's very well known that, like I said earlier on, that in some special instances, when the individuals initiate therapy early and interrupt therapy, they can go in remission. But those are exceptions. In most cases, when individuals get off therapy, they rapidly rebound. So in the field, the question has been, where does the virus come from? This is an example in FRESH. Somebody was treated in phoebic stage 1 and they were completely suppressed in terms of viral load. They missed the medication, the virus quickly came back, they had to be put back on therapy and then were suppressed again. So where does this virus come from? We know that virus tends to hide in what we know as tissue sanctuary sites. These include mostly lymphoid tissues, maybe the brain, the testes, and other tissues of the body where the immune system is suboptimal, and then the drugs may not penetrate very well. So if we are going to develop a cure, we have to understand what's going on in tissues in individuals that are on therapy. And so part of the efforts of my lab has been to create what we call a tissue biobank. So this is a program where I've been collecting excisional lymph nodes, tonsils, gut tissues for research purposes, some from surgical procedures, clinical procedures, and some just directly as a research material that we get from volunteers. We've also been collecting mucosal samples, and from the hospital, we've been collaborating to get bioresections, and plans are underway to get postmortem samples as well. So for the purposes of my talk today, I'm gonna focus on the lymph node samples that we've been obtaining from some of the girls that have been participating in FRESH, that we consent to donate a lymph node for research purposes and there have been, some have been, kind enough and generous enough to donate an excisional lymph node. So we've collected lymph nodes from these individuals at different time points during the course of infection and during the time they started therapy, from like within a few days of initiating therapy, and some we collected lymph nodes after years on therapy. So here I'm just giving an example of somebody that we studied in the fresh cohort that was suppressed within 2 weeks of diagnosis, and we only managed to get a lymph node after 1 year on therapy, and they had no blips in terms of plasma viremia whatsoever. So when we looked for HIV antigens by immunofluorescent staining, here we stained GAG-P24 with green in this tissue, lymph node tissue section. You can see that we see this green foci of staining, which is GAG-P24. And when you zoom in here, we counter stain with BCO6, which stands for active germinal centers. You can see that the GAG-P24 staining in green is confined within this germinal center. We've gone one step further and also tested for HIV RNA using RNA-scope, using either immunohistochemistry or immunofluorescent microscopy combined with RNA-scope so that we can multiplex it and identify cells that harbor the HIV RNA. So on top here, this is the negative, which is our control. You can see we can detect in amphibic stage 1 individuals, in late treated and untreated HIV RNA. Of course, there's more in untreated and late treated compared to early treated, but detectable nonetheless. When we multiplex with CD4, we can see that most of the RNA is confined within CD4 positive cells. And here, just showing you the quantification of HIV RNA. And when we compare HIV GAG-P24 staining and HIV RNA in these individuals, we see a very nice correlation suggesting that, you know, the individual's transcription might even be leading to this HIV protein that we're detecting by GAG-P24. We've gone one step further and tried to sequence the virus in the lymph node. First of all, these individuals that we study are put on therapy within 24 hours of detection. When we sequence the virus initially, you can see they are infected by a single transmitter founder virus. And so we do not allow the virus to diversify before they are put on therapy. And so it's easier for us when we look in the tissues and we see that the virus are diversified, suggests to us that there's ongoing virus replication. This is an example here where a highlighter plot where you see a HIV envelope sequence in the lymph node, very different mutations in terms of compared to the sequence in the plasma before therapy. We've done a few other patients in GAG and have identified mutations that are confined within non-CTL epitopes restricted by these individuals' HLA alleles, suggesting that there's ongoing immune pressure, at least CD8 T cell pressure, that is leading to these mutations. So we believe that a combination of HIV GAG-P24 staining that we've done and identified HIV GAG-P24, the RNA scope, which suggests that transcription is going on, and the sequencing suggests that at least in some instances, there is some low-level ongoing virus replication in lymph nodes, even when you treat people that early. So the next question has been, can we define the phenotype of these cellular reservoirs? Because then if we can identify the cells that are really carrying the reservoir, we can specifically target them for elimination. So here I'm showing you an image of a lymph node stained, GAG-P24 in yellow, the BCL6 in green, and CD8s in red. You can see that for the most part, the CD8 T cells are excluded from the germinal center where the HIV is persisting. So we wanted to find out whether there is a biomarker for cells that support virus replication and harbor latent reservoir. And so we've been doing a lot of staining using different markers, PD1, BCO6, CXCR3, CCR6, and follicular dendritic cell markers to try and understand which of these cells markers co-stain with HIV GAG-P24. And we found several markers that do that. And then we've sorted cells based on different markers and measured HIV RNA by digital droplet PCR. What we found is that we detect HIV in different CD4 cell subsets, but mostly we see a significant increase in HIV RNA in CXCR3 positive CD4 T cells compared to the other subsets, suggesting to us that this might be a marker for the reservoir. We've gone on into the lymph node and stained for HIV-specific CD4 T cells using a BNAB, which stands for HIV envelope, and look to see the phenotype of these cells without any prior manipulation. And we've seen that there's a significant increase in CXCR3 positive envelope positive, HIV envelope positive CD4 cells compared to CXCR3 negative. So combined, these data suggest that CXCR3 might be an important marker for the reservoir, although it's not exclusively the only marker for the reservoir. So lastly, because of the limitation with our microscopy, because we can only do 4 colors and we cannot resolve really the subsets in greater detail, I started a collaboration with my colleagues at Stanford University, Michelangelo, who has developed this high-tech imaging platform known as MIBI-TOF, which is multiplex ion beam imaging by time of flight. So this is a mass spec imaging technology where you use antibodies conjugated to metal detectors. And the advantage of this technology is that there's no spectral overlap, and so you can stain for as many markers as you wish for and define things in much greater detail, even at subcellular resolution. So we've sent some of our tissues to Michelangelo's lab at Stanford, and they've done MIBI staining on some of our tissues. And just to give you a flavor of what we are able to do is you can stain for all these different markers in one staining, and so you can get very detailed resolution. And this is a MIBI image that we image one of our lymph nodes that has HIV GARP24, and then we can begin to define the immune microenvironment around the reservoir at subcellular resolution. So we are extremely excited about this technology, and it's going to tell us a little bit more about what is happening in these lymph nodes. For instance, one of the studies that we've been pursuing is this scenario where you have within one lymph node, you have some germinal centers that are chock full of HIV GARP24, suggesting that the reservoir is right there. And then you have these other GCs within the same tissue that do not have HIV GARP24. The question becomes, what is the immune microenvironment around the reservoir versus where there's no reservoir? Can we identify the players that are actively influencing the reservoir? So these data are very preliminary, but what we've done is we examine the tissue composition, cellular composition in the area where there's GARP24, here in GC1 and GC2, compared to an area where there's no GARP24 in GC3. And you can see just by eyeballing that the cellular composition in the GC1 and GC2 look very similar, whereas the cellular composition is different in an area where there's no HIV GARP24. And so what we are hoping to achieve is to try and identify the key immune parameters that are in and around the reservoir and try to understand, of course, we have to unpack the chicken and the egg, whether they're actively involved in influencing the reservoir or it's a consequence of the reservoir that creates that environment, is something that we are pursuing very actively. So for this part, the conclusions are that HIV GARP24 antigen persists almost exclusively in the germinal centers in early treated people, and we can quantify HIV RNA in these tissues in early treated individuals, and CXCR3 positive germinal center T follicular helper cells seem to have a greater HIV RNA in early treated individuals. And I've just shown you just a snippet of data that demonstrate that this technology maybe can be used to image the immune microenvironment around the reservoirs in fixed tissue, human tissue. Lastly, very briefly, let me just touch upon some of the work that we are doing on the CD8 T cell front. What we've found is that we see this heterogeneity in people that are infected with HIV and are treated in phobic stage 1, ranging from almost no detectable HIV antigen to detectable very high HIV antigen in spite of initiating therapy almost at the same time. So we've looked at the immune responses in individuals with little to no HIV antigen detected in the lymph node compared to people with a lot of HIV antigen detected. And we looked at their CD4 T cell responses by interferon gamma here from the cells derived from the lymph node. We found that the individuals with low antigen burden had better interferon gamma secreting CD4 T cells compared to individuals with high antigen burden. And these are the summary slide. And similarly, we see the same result with the CD8 T cells. Increased interferon gamma secreting, persisting interferon gamma secreting CD8 T cells in individuals with low antigen burden compared to high antigen burden, suggesting that CD8 T cells might be playing an important role. But the issue here is that the CD8 T cells, when you stain them in the lymph node, they are almost excluded from the germinal center, where I've shown you in several slides. That's where the HIV persists. So us and others have been trying to figure out how can we get the CD8 T cells into the GC to eliminate HIV. So for the CD8 T cells to enter the germinal center, they need to express CXCR5, which is a chemokine required to enter the germinal center. We've looked at the expression of CXCR5 positive cells in untreated HIV infected, treated, and HIV negative. We see that in HIV infection, there's an increase on CXCR5 positive cells, as you can see here by imaging also the yellow cells. In HIV negative, you can hardly see any CD8 T cells in the GC. In HIV treated, you see some CD8 T cells going into the GC, and in untreated infection, you see even more CD8 T cells going into the GC and expressing CXCR5. What about HIV-specific CD8 T cells in the lymph node? So we can identify them with tetramers and look to see what is the proportion of these tetramer-positive cells that express CXCR5. And you can see it's a very small and low expression CXCR5, suggesting that, these HIV-specific CD8 T cells have a hard time going into the germinal center. And so I just received grant from the Gates Foundation to develop novel strategies for inducing CXCR5 expression on CD8 T cells as a way to increase their trafficking into GCs using small compound screen strategy, and also using CRISPR-Cas technology to silence CXCR5 expressive genes as a way of engineering CD8 T cells to go into the germinal center. So to conclude this last part, I've shown you that CD8 T cells do not generally enter GC lymph node follicles unless they express CXCR5. And in the lymph node, CD8 T cells express reduced levels of cytotoxic molecule. I didn't show you those data required to kill cells. And then we've also generated data that implicate DNA methylation as a way, as a molecular mechanism by which CXCR5 is repressed on CD8 T cells. So if we're going to eliminate HIV, we have to figure out how to clear HIV from the lymph node tissues, and it has to be an integral part of any HIV cure effort. So lastly, I'd like to thank all the people involved in this work. From my lab, my postdocs, my PhD students, Omarala and Funsho, did most of the work that I showed you today. And Jacqueline is a collaborator, and Tumbi is also my collaborator that contributed to the sequencing data that I showed you today. And a fresh cohort run by Krista Dong and Amber, and the fresh participants have been key to recruiting our study patients and making sure that we're able to get these samples. And of course, the hospital, the surgeons that help us to get these lymph nodes. And at Stanford, Michael and Candice and Erin did the MIBI work. And I'm also funded by the HHMI, NIH, and the Bill and Melinda Gates Foundation, IAVI, and the Reagan Institute. And thank you very much for your time and for listening.
Thank you, Zaza. This was spectacular. Really amazing your work and figuring out these sanctuaries where HIV reservoir persists. Yes, thank you so much. I have some questions, but I will ask them via Twitter, as Carla will indicate now.
Thank you. Yes, thank you, Zaza, so much for sharing these fundamental new understandings on such an important infectious disease. And thanks for your efforts to train the next generation of South African immunologists. It's phenomenal. So I just want to remind everybody that Zaza will be answering the questions via Twitter. There's already a tweet that you can find in our Twitter account. And please remember to reply to this tweet, and Zaza will be using his own Twitter account. So we look forward to your questions. Zaza, thank you again. And we look forward to seeing everybody again next Wednesday with Richard Loxley. Thanks, everybody.
Thank you, Zaza.
Bye.
See you, everyone.